Thank you very much, and hi everyone. And I will talk a bit about how we can use precision medicine and network medicine in ME research. And I will still show some networks from IBD, which I have used these particular techniques, which we can hopefully will move forward and use them in ME as well. So, little problems. I have nothing to declare. So just like what we have heard in the last few days, in ME research there are so much data nowadays. Last year when I have came here, there were not as much big data sets like what we have now. We have the health records ready. We have much more high throughput omics data which we can use. And we can use that with artificial intelligence and systems biological knowledge to hopefully improve the prognosis, understand the pathogenesis, profile of patients, that you understand them a bit better, and maybe find some kind of biomarker which we will actually decide that which particular drug we can use in ME. But why to use network biology? When you have your data set and you find some kind of single nucleotide polymorphism or some kind of mutation or effect of a virus, then it's usually not just directly affecting one particular part of the system, but it propagates through. And with that, we can get actually enrich our existing knowledge. With that as well, we can understand a bit better. We can find that what pathways we will have. If you will just use transcriptomics with for example, in ME, and we don't see much changes, especially when we have just looking at the normal situations. We have to a bit more go back and see that what are those little changes which are maybe a bit different and uncover some hidden variables. How we can use these biological networks? We can, in my study field, we usually use two types of biological networks, either the protein-protein interaction networks or regulatory networks. In PPIs, the nodes and vertexes are usually the proteins. The links which are connecting these proteins are physical interactions. They are described, they have been measured by some kind of biochemical laboratory. Then we don't know that how is it directed or indirected, these interactions. But if we do it well, we can move them directed. We can check that whether they are signed. Signage meaning in this case that whether it's an activatory or an inhibitory interaction. PPIs can be signed as well. When we will get the graph, which is the summary of all of these interactions, then we will get a normal graph. So we will have both of the type of interactions will have the same level. All the proteins will have the same level. And there are many databases, for example, the OmniPath or the string database, which I am usually using when I am building these kind of protein-protein interaction networks. The other thing which we can use is some kind of regulatory networks. In that case, we have some kind of transcription factors or microRNAs which will have target genes. So if we will look at here, we have immediately see that we have two types of nodes here. One of the type which is regulating, and one which is regulated, so that we have a transcription factor which regulates its target genes, which means that the links are regulatory interactions, and in this case they are directed. And in this case, we can know that whether that particular transcription factor will have a positive interaction for the target gene, or in a microRNA case, it's usually a negative interaction for the particular target gene. This kind of graph is so-called bipartite graph because the transcription factors will have a target gene, and these are one-directional interactions, while the transcription factor will have only one, or only can regulate to a particular target gene. There are many databases as well which we can use for that. For example, the Dorothea database for transcription factors and the target scans, for example, microRNAs. And when we can join these together, then we can work on it, that how we will build biological networks. A big basics of artificial intelligence as well. We can have 3 different types of artificial intelligence. When we use them as a supervised learning, we have some kind of information which we know, and then we want to use the AI to, hey, these are our ME/CFS patients, and we want to diagnose them from these unknown patients, that whether they are also ME/CFS or not. We can have unsupervised AI. Hey, we have here all of our different type of ME/CFS patients. Do we have some kind of clusters in there? Do we have some kind of different type of AI, data there, which they define the type of patients which we have? Or we can have generative AIs. These are that we have, which you hear about all of the time, the mid-journeys and similar stories. So in supervised AI, we can have classifications or regression problems, basically define that which one will be the type of whether they are ME/CFS or not, predict something like some kind of disease severity score, for example, and it can help to the decision making. We need a very metadata-rich data sets. Here are some typical algorithms, random forest, nearest neighbors, SVM, VM, etc., etc. These are the methods which we are usually use for this kind of stories. Unsupervised machine learning algorithms, they find the hidden structures in your data sets. It works without metadata. We are basically generating that metadata data, but usually there is no best structure. If we have seen, for example, a single RNA-seq data set, when we have looked into there we just see the different clusters, but often we need to figure out that what those clusters mean by investigating, and then deciding that whether we should be fine with that kind of level of different clusters, or we want to go, for example, a bit deeper. Typical methods, hierarchical clustering, k-mean clustering, or we can embed them with neural networks as well. Generative AIs are generating new data from existing ones, so like Midjourney and other types, creates brand new data points based on learned parameters. And sometimes they can create false outcomes. Where we can use that in ME/CFS, that we can basically, from the existing ME/CFS pool, we can generate additional patients, which are more maybe similar to what we have as well in a digital twin form, basically. And then from that we can use for additional measurements. The very important thing for every AI and every machine learning story, if you have bad data, garbage in, you have your fantastic, super awesome model which gets including every little biology. It doesn't matter, it will be still garbage out. So you have to be very much careful that what you will put it into your data set. So I will now go to my field where I have used this kind of methodologies. And here we have can we decipher the pathogenesis of IBD using only SNPs, and in the one type of particular IBD it will be Crohn's disease. Crohn's disease is an autoimmune disease. It's one of the IBDs, and we still don't know the pathogenesis of it, just like in ME. We have important thing that we have here, these little transmural infections of the bowel wall, and we know that only 30% of the patients can get treatment, and it's also increasing. So here we have something which will cause an effect, and I will follow the ripple of that particular stone on the surface of the water. And that's how I will try to understand a bit more of the IBD. The stone in this case will be single nucleotide polymorphisms. Here I had quite a lot of them, thanks to the IBD clinics in Leuven. And I am looking at that whether these single nucleotide polymorphisms are in the transcription factors enhancer or promoter region. And when they are there, these SNPs are creating a new one of this transcription factor, or make these transcription factors not be able to bind to that particular promoter. And I can do the same thing with microRNA as well. Where there is a single nucleotide polymorphism, then that microRNA will maybe not be able to bind, or there will be a new microRNA which will be able to bind to that particular place. And when we have that, then we can go to find the SNP-affected genes. From that, the SNP-affected proteins, which we can move to a specific network. And then we just follow the signal, follow the ripple effect over the water, and then go down, 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 till we will reach the transcription factors. And then one more step, and then we will get some kind of SNP-propagated regulatory network. And when we will have that, then we can check that whether this makes any sense. So we will get some kind of functional analysis or patient-specific pathomechanism. Hopefully, because we are getting there, we can basically do that as well with ME/CFS. In IBD, what I have done, it looks like something like this. This is the signaling network of Crohn's disease. Each of these groups are related to such part of the network which is commonly affected. These are so-called modules, which means that they are more interacting within each other than outside of them. And we can find very common pathways which are involved in this network. Apoptosis, interleukin-2 regulation, immune response, for example, in this one, JAK-STAT regulation, cell cycles, DNA damage in this one, and cell migration in this particular place. And we have found even autophagy, which was one of the common problems in Crohn's disease, which is a very well-known cause of it. So this is what we can get from a network. Again, we have figured out some little hidden variables as well. Especially if we will move one step further, when we will go from the transcription factors and their target genes in the gene regulatory network, we can find some very nice, for example, these ETS1 and ETS2, which were potential drug targets even in IBD nowadays, and we have been figured them out as well, correlating with a paper recently. And the BRF, STAT3, etc., etc. So it's very big transcription factors. And then we can check as well that what they are involved. For example, the MAP kinase signaling, neutrophil degeneration, integrin cell surface, etc., etc. So when we have that, then we can look at that how each of these patients, so each of these dots are one patient, and then we will use the machine learning and do the clustering. That's a very tough choice to decide that how many clusters we have. Based on of my measurements, I have decided that we will have 12 clusters because, well, the two measurements, the average silhouette score and the inertia, was more or less that where we had here an elbow, and here it was higher than what we expected, and I was not happy with only 4 clusters. So based on that, we can get back that which transcription factors are involved indirectly. And from that, we can get for each patient these particular signaling networks. So each of these will have a particular transcription factors and target genes which will be affected in the various pathways. And when we have that, then, well, you can say that this has beautiful network stuff, but it has not much meaning. You can check that when you have some single-cell RNA-seq data and corresponding that, whether the differentially expressed genes in various types of single-cell RNA-seq cell types corresponds with these clusters. And if they do, then you can say that in certain clusters, that's what you will get back. So this is what you can do in IBD. So can we do something similar, follow the ripple effect of the stone in ME/CFS? I really hope that next year I will come back and I will show you that yes, we can. I cannot do that yet, but I really love that in these few years there are ready single-cell RNA-seq data and RNA-seq data which we can use for this. So we have some kind of source nodes which we have in ME/CFS. We have seen that there is maybe a viral trigger, maybe differences in the TCR receptors, immunoglobulins on the MHC factors are working slightly differently. We can, from that, follow downstream in the signaling networks. If we can find the regulating transcription factors, and then we can connect that with transcriptomic response. This is one way which we can do that. Or we can follow the ripple, which will be the transcriptomic response, and follow that backward. For example, the single-cell RNA-seq results which we have seen today. And we can find that what are the which are regulating those transcription factors. We can go back and what are the signaling networks which are there. From that, we can guess that whether these proteins are actually involved into the viruses. And from that, we can figure out that if we do that in patient-specific manner, what is the potential cause which we have seen in that particular patient, if we have here something which is connecting. That's how we can use networks to give a slightly better understanding in ME/CFS, and also a slightly better understanding and a slightly better clustering maybe of patients. So systems biological methods can uncover hidden pathways in complex diseases, and I have done and hopefully demonstrated for you that we can get that from patient-specific clusters which corresponds to various cell types, and we can maybe stratify with them patients as well, and we will need a large amount of clinical metadata to do that. That was one of the, and still my pain, that in IBD we didn't get access to that. So if we will be able to do that for all of these various interactions in ME/CFS, do the networks together and build up cell and condition-specific interaction networks, use the machine to do unsupervised machine learning techniques, then we will be able to get patient-specific pathomechanisms and disease clusters, just like what we have got here. And then we will get a bit better understanding. And I could not work, of course, without my colleagues for that, and thank you very much for your kind attention as well.